Good afternoon, everybody. Welcome back. I think the only qualification I have for chairing this session is I'm the only one in the building with a tie on, so that's the reason. Anyway, I'd like to introduce this afternoon Elisabeth Rieper from National University of Singapore Center for Quantum Technology. She's going to talk about classical and quantum information in DNA. Thank you.
Hello, everybody. First of all, I would like to thank Google for organizing this really cool workshop. And I will talk a lot about information, and going to Google and talk about information feels very interesting. But okay, I know a little bit about quantum mechanics. So first of all, I will spend a couple of slides to explain the title in detail. Then very briefly, I will talk about the coherence. Then all those concepts developed in the first section will be applied to DNA. And then the really interesting part is, does quantum matter? I can say it right now, I do not know, but I will make some speculations. And then finally, I will compare those different computing structures like classical computers, DNA, and quantum computers, and show where they are similar and where they differ. Quantum biology suffers from one severe problem, namely that biology is a massively complex system, whereas quantum mechanics is massively deep. So if you change a little bit, like we've seen with the talk from Luca Turin, suddenly everything changes. So combining this complexity with the deepness is a challenge. So for combining quantum information concepts with DNA, I had to do some simplifications. And I apologize to any biologist if those simplifications turn out to be too brutal. Details really matter. So what is DNA? Let's start with the Wikipedia definition of that. So it's a deoxyribonucleic acid. So that's a nucleic acid that contains genetic instructions used in the development and functioning of all living organisms. The main role of DNA molecules is a long-term storage of information. So the last sentence, that sounds rather innocent, actually contains two very interesting concepts. So the first concept is information, and the other one is long-term storage. So what does it mean, long-term storage? It means that we have some information, whatever information is, in the past, and we would like to send this information into the future. That's long-term storage. And the way we send it from past to future can be described by a channel. And a channel is a very general description of whatever happens to your information. So what is information? Well, I already asked this question to Google. That was the answer. It came pretty quickly, and it's got a lot of results, but I actually did not bother to read all those results. So as a consequence, I will give you a couple of my own definitions, what I like to think about information. First of all, you can see it as negative entropy. So now I run into the problem that I have to explain you what is entropy. Entropy in general measures the ignorance you have about the system. If your system has zero entropy, you know everything about it. You have maximum information. If your entropy is large, there are certain things you do not know about it, and you have few information. And entropy is a well-established concept in information theory. So how do you use it? You choose an alphabet. That could be computational basis of 0 and 1 or anything else. Then in the next step, according to that alphabet, you count probabilities. And once you have that probability distribution, you can calculate the entropy of your choice. There's a whole family of it. Here I've shown you the Shannon entropy, which is widely used and very useful. But I'm a physicist. We can do the same in statistical physics. In that case, we choose our system of interest, and then we count the number of possible states, which is denoted by capital omega. And then we take the logarithm of that number, and KB is a constant. Don't care too much about the meaning. The massively interesting thing is that once all events are equally likely, those two definitions of entropy actually coincide. This is one of the many links between information theory and physics. So it's rather a combined science than something different. And in physics, we have the well-established second law of thermodynamics, and that tells you that in closed systems, entropy does not decrease. So if we translate that argument to information theory, we immediately know that in closed system, information cannot increase spontaneously. That's very important to know. Next thing I would like to point out that information is physical. Whenever you have a piece of information, it comes together with a physical carrier. That can be the piece of paper you are writing now at the moment, or it can be your hard drive on the computer. And for classical physics, for classical information, we know from everyday life, I can change that information carrier. I can write something on a piece of paper, I can scan it to my computer, and I can print it again. And if I didn't do obvious mistakes, I didn't lose any information. That's allowed by laws of classical physics. But as the information comes along with a physical carrier, the way I process information is bound to the laws of physics. So what happens if I choose my physical carrier to be smaller and smaller until I reach the quantum limit? Then the fascinating thing is that the way I process information changes according to the laws of quantum mechanics. So what does it mean? So one thing is that I cannot copy quantum information. I can only copy classical information. And whereas classical information reliably stores the information, in quantum mechanics, I always have to find the coherence. It's always getting lost. So coming back to the problem of DNA to use a long-term storage, what I just told you, those 2 points would actually mean that quantum mechanics is not any good for DNA. And if you really want to store your genetic information safely, you should keep away from quantum mechanics. So what is a good point of it? Quantum mechanics allows you to do more. Classical mechanics is a special case and only has a certain set of manipulations, and quantum gives you more. So now the question is, does nature exploit this edge of being able to do more? Just a very short reminder of the notation. If you have a classical bit, that's usually denoted by 0 or 1. If you have a quantum bit or a qubit, you put the 0 and 1 in these brackets. We call them cats. And they have one interesting feature is that they allow superposition. So in the classical case, you have to decide whether the qubit is 0 or 1. But in the quantum case, you can combine it in a superposition. And then you have a mixture where the qubit is either 0 or 1. You just don't know which one it is. So in the superposition, it's both simultaneously, and in the mixture, you don't know which one it is. So I told you that you cannot copy quantum information. The proof is a 3-liner. So let us suppose you could copy quantum information. Then you would have some unitary acting on your wave function psi that actually copies an unknown state to your template. And then I can suppose that state psi is a superposition of 0 and 1. And I plug it into the formula, and I get that lengthy expression of the copied state. But the mathematical formalism of quantum mechanics also allows me to feed it in step by step. So I can feed in first the beta cat 1 part, which is copied to 1, 1, and then the alpha 0 part, which is copied to alpha 0. And that should be the same, but we can see clearly it's not the same. So our assumption that we can't copy quantum information was actually wrong. And that's called the no-cloning theory. Actually, there was one special case where these two lines are the same, namely if either alpha or beta are 0. And this tells you which would roughly correspond to classical information, so that's something you can copy. Another thing I would like to mention in my introduction is conditional entropy. So the conditional entropy measures how much uncertainty, how much ignorance you have about a combined system and observer, given that you have all the observer's knowledge. And for anything that we encounter in the real world, it's a fair assumption that this conditional entropy is bounded below by 0. So the idea is that if my observer has a certain ignorance about his own state, then the combined system and observer should have at least this uncertainty. So, but if we are now dealing with this state, as was mentioned before this morning, actually I have full knowledge about the full system. Globally, I do not have any uncertainty. But if I look at the state the observer himself has, he actually has uncertainty. So this quantity is actually, for quantum systems, this conditional entropy can actually be smaller than 0. And whenever something like this happens, we call it entanglement. So whatever physical meaning entanglement has, you can take it as very strange probability distributions which allow you to achieve correlations which are classically not possible. So one application. So let's suppose you have a system which is in a mixture of 0 and 1. And you would like to reset. That could be, for example, your memory. And you would like to reset that to state 0. And what is usually called Landauer's erasure principle states that a classical observer has to pay 1 unit of work to extract or to erase that information. Is it any difference if we are dealing with a quantum observer? Yes. It could be that the quantum observer actually has the other half of a maximally entangled state. And then the system has the same correlations. And as was shown recently, in that case, you can actually extract 1 unit of work and erase the information to reset it to 0. And now we are talking about biology. Whatever small enzymes, molecules, and other systems are wiggling around in your biological systems, it could be that they are small enough to be a quantum observer. And then suddenly the whole setup changes. Instead of needing to pay work, you can actually extract work and do the same. So it might be that for those small biological systems, we need to change the way we think about it. And yes, another thing I would like to mention is information is a subjective quantity. It depends on the knowledge you can have, and that might be different for different observers. So this was the introductions with a lot of concepts. That's the only slide I will mention about the coherence. You can talk a lot about it, but I will abbreviate it to the time a system can maintain its quantum coherence. It's roughly inverse proportional to the mass and the temperature. So why don't we see any quantum effects in the real life? We are massive. We have a lot of mass. And the second reason is we are hot. We are at 300 Kelvin. That's a lot of temperature. So whenever you have these two effects, you would believe that you don't have quantum effects. So finally, information in DNA. So probably most of you already know this molecule. Let me just give you a brief reminder. So this DNA is this really thin and long molecule. Outside, you have the phosphate backbone and the sugar stabilizing the DNA. But it's actually those 4 nucleic acids that contain the genetic information. And when I first looked at those molecules, I thought, well, they are pretty small compared to our computers, what we encode in a bit in a computer. And because here we have 4 different bases, each molecule encodes 2 bits. And then these 2 bits are arranged in a linear array, and so the 2 strands have opposite information. Now the interesting question is, how do you actually access that information? It's not enough to store it. You actually have to be able to read it out. How do you do that with these small systems? So let's look at only one of them, cytosine, for example. So that's for you the right side, isn't it? No, that's for you the left side. So on the left side, it's actually bound to the phosphate backbone, so you cannot access it. Up and down of this cytosine, you have electron clouds and other nucleic acids. So you cannot access that part either. So the only part where you can actually access that molecules is on the right side. And this is actually what's used in biology. Here we have those atoms, that's nitrogen and oxygen, and single protons can bind to it. So now the idea is to use the position of those protons to encode the identity of that molecule. Another thing I should mention is that this aromatic ring you see there, that's actually, even though I consider it small, for quantum mechanics it's actually quite large. So this is something we can safely assume to be classical information. Whereas the position of a single proton is due to laws of quantum mechanics. That might tunnel. So this leads me to this notation. It's a little bit sloppy, but it's good for transferring ideas. So the classical information that we are dealing with the molecule cytosine is something I would encode in this quantum state, which means that the lower 2 slots where the proton could be are empty, and the upper slot is filled. So that's denoted by those zeros and ones. So then how do you combine it? Cytosine always pairs with guanine, and so guanine has the opposite slots. So the lower 2 slots are filled, and the third one is empty. And then when you bring those 2 molecules together, you can form H bonds. And you can only have 1 proton in each H bonds, so that's why it's necessary to have this sort of matching proton distributions. So here it's really a lock and key mechanism. Same holds for thymine and adenine. So does this mechanism work perfectly? No. What can happen is that your cytosine goes into its rare form, which means that its proton tunneled to the second slot. That's allowed due to quantum mechanics. But then when we go to the lookup table of the encoding, this actually looks the same like a thymine. And as a consequence, because the quantum information pretends that this molecule is a thymine, the adenine thinks that it can bind to it. And that's one way how you can get mutations to your system. So once again, you have your classical bit, which you encode in this quantum information, and epsilon is some sort of a tunnel probability. So epsilon is small. So most likely it stayed in this state, you encoded it, but there was a small probability that your quantum information was destroyed into a different state. So this is quantum information in the transversal direction. So let's make a little jump to the longitudinal direction. All these nucleic acids, they are more or less planar molecules being surrounded by electron clouds. These electron clouds, because you're dealing with aromatic rings, are highly delocalized. So that's why I draw them in such a sandwich notation. So now it happens that these electron clouds carry negative charge. And we know that adjacent negative electron clouds will repel each other. The intriguing thing is that this actually leads to correlated excitations lowering the ground state energies. So different charge clouds kick each other, and then they redistribute their charge distributions, and that can actually lower the ground state energy. Now, that effect is called van der Waals interaction, or van der Waals forces. And you actually need the coherence between neighboring sites to get the attractive part of the force. But now this also has non-trivial implications. So first of all, neighboring sites are entangled due to this continuous interactions. And also, the electron cloud of one side carries information about the identity of the other side. So if I'm a thymine here, and I'm adjacent to an adenine, this adenine influences my own electronic state. If there would be a cytosine next to me, my own state would look different. And that's substantially different from classical computing. So my own identity is influenced by the neighbors I have in the DNA chain. Now, I just gave you two examples where you can expect quantum information to be present in DNA. And maybe the one or other just was irritated because we are room temperature systems. So how does that work together that I told you if you have massive hot systems at room temperatures, you cannot have entanglement? Well, I didn't tell you everything. Details matter. So you can analyze a very generic model of coupled harmonic oscillators. And then you actually see that the absolute temperature does not matter. It's a temperature, so kBT, the thermal energy, compared to the interaction energy that matters. And if that coefficient is small, then you can expect quantum effects to be present. And if you plug in all the numbers we have for this longitudinal entanglement, you see that thermal energy is actually very small compared to the interaction energy. So DNA has optical frequencies, and this means that the thermal noise cannot excite those optical frequencies. So this quantum information we are talking about is basically in its ground state, even at 300 Kelvin. So here is a screenshot from a computer program where you do something with DNA. You've seen it many times before. And to sum it all up, what I told you in the previous slides, everything that you can see on this picture is classical information. The black stuff in the double helix that you cannot see, that's a quantum information. So I haven't discovered any good way how to picture correlations. Probably there is no way, so that's why nobody ever draws quantum information. But the next time you see such a double helix, you know, okay, all the stuff I do not see, that's a quantum information. So basically what happens, this classical information, which is usually drawn here, is embedded, is sandwiched into quantum information. And most likely, this classical information is never accessed. You can only, because it's inside all the quantum information, you can only access the quantum information, or the electron clouds and the protons. So mathematically, you can describe that as a quantum classical state. So let's suppose that X is a string of classical symbols. You would denote that the state has two components. So the first one denotes the classical information. This is diagonal in its basis, has no coherence. But this actually comes along with some sort of quantum information. And so X, for example, could be this string of your DNA. And then it is embedded in this cloud of quantum information. And Paul Davies calls this sort of quantum information shadow information. Because in this quantum information, you have at least one copy of the information in your DNA, if not more copies. And if you want to decode your classical information, you measure the quantum part. But it can happen that the quantum part of sites is not fully distinguishable, and this gives possibility for mistakes. So coming to the most important question, does it matter at all? Well, I do not know for certain, but I think there are many things quantum allows you to do, and if quantum gives you an advantage, this 4 billion R&D project probably discovered how to use it. Let me come back to the channel picture I introduced in the beginning. So you would like to send some classical information from the sender to the receiver. We do it every day. We send text messages, we send email, we talk. That's also classical information, which uses classical channels. The only problem is that this probably doesn't take place in DNA because the system you are talking about is too small to really have a full classical channel. So it's more likely that you're dealing with a quantum channel. You can send classical information in a quantum channel. That's no problem. So you encode your classical information on your quantum system, you send your quantum system, then you make a measurement on it and decode the information it carries. And there are two channels I would like to compare. So the normal quantum channel and the entanglement and sister quantum channel, where actually the sender and receiver initially share entanglement. And then I will talk about the capacity of channels, and this roughly measures how much information can be transmitted in this channel. So that's a number between 0 and 1. If it's 1, you can fully transmit all the information you send in. And if it's 0, your channel is useless, and you cannot send any information. Calculating channel capacities is very, very difficult. And those capacities are not known, but for some simple channels they are known, and one of them is called qubit depolarizing channel. So what happens, you take your wave function psi of x, and you send it through the channel. But it can actually happen that in this channel it suffers from some sort of noise, as we can expect it with biological systems. And then with probability 1 minus epsilon, this channel gives you the original wave function, and with probability epsilon, it sends you something completely random. So that's a qubit depolarizing channel. And now, if you would like to encode your classical information x, so x could be adenine, thymine, or whatever, you encode that in a certain wave function. You send it through your noisy channel, and with some probability, you recover x. But in some cases, you guess incorrectly, and you recover y. So how often you guess correctly and how often you guess incorrectly is characterized by this channel capacity K1. And now you can use the same channel. The only difference is that now sender and receiver initially share maximally entangled states. And then you have the capacity KE, E for entanglement. And the beautiful thing is, if you use entanglement, you can double your channel capacity. So we have a system where you would like to transmit classical information. It is reasonable to assume that you can create this entanglement resources, and this entanglement would allow you to double the capacity of your noisy channel. So that's something worth thinking about. Something completely different, how you might use entanglement in DNA, is for communication along the chain. When I read up on the literature on how little motors go along the DNA and achieve things, I always wonder, but how does it work? How do these motors actually know what they are supposed to do? So let us suppose that each of these balls represents a site in your DNA. And what I do is I wiggle the one end of it. And then you can calculate what happens to it. Actually, after some time, you will get entanglement between the first side where you shake your end and the nth side, could be the 10th or 20th or 30th side along the chain. And suddenly, these two far apart sites show entanglement. And entanglement means that they have correlations. So the question is, can you use those correlations for communicating along this one-dimensional chain? And the next thing is something where my own ignorance comes up. I do not know too much about protein folding, but I have a background in information science, so I would like to ask a question to people who know more about biology. So when I would like to fold a protein, I start with my DNA, which is a one-dimensional sequence of information. So in order to fully describe the classical information in DNA, I only need to know this one-dimensional sequence. Then I make a copy to RNA, and the information content did not change. In the next step, I translate pairs of 3 of my RNA into an amino acid. Now, there's some sort of code on code. You might argue that you lost some information. I'll ignore that effect for the moment. And then in the 4th step, this one-dimensional chain of amino acid actually folds into a 3-dimensional structure. So let's look at it again. You start with DNA, messenger RNA, and this chain of amino acids. And because they are all linear arrangements, I would argue that roughly, up to small errors, they carry the same amount of classical information. And then you fold the protein, and it's getting a 3-dimensional structure. And the shape is vital to its functioning. But now you need to store more information. In addition to the sequence, you have the position information. And in my opinion, that's actually an increase of information. But we also know from the second law that in closed systems, information cannot increase spontaneously. Now I know that creating a protein is far away from being a closed system. But my question to the biologist is actually, where does this additional information come from? If you think about it in detail, that's actually not that trivial. Does it come from the time sequence in which you assemble the protein? Does it come from some sort of laws of quantum mechanics? Do you have some sort of quantum information actually also taking place? If anybody can tell me, I'd be very happy. So a Gretchen question that's actually taken from a German piece of literature, Goethe, Faust. It's a very nice book. I can only recommend reading it. So it's sort of an important question. So the important question is, does quantum information matter? And there's one way to test it. So you take 2 identical strings of classical information. And by some means, in one string, you change the quantum information. There are some ways to disturb quantum information. And then you actually look, does it fold in the same configuration, or does it fold differently? If 2 strings with the same amount of classical information but different quantum information fold into different configurations, different shapes, that would actually be a good argument that quantum information matters. And this line of thought generalizes to other situations. When you wonder, does quantum matter? Well, disturb it, shake it, destroy it, and see if it makes a change. And then you know if quantum matters. So last point, comparing those different platforms which all have to do with information flow. If I have a classical computer, I would initialize it in either 0 or 1 or any other computational basis. For DNA, I argued that you use quantum classical states. And for quantum computing, I didn't talk too much about quantum computing, so if you are unfamiliar with the subject, you can just ignore the last column. You have whatever space your quantum computing is assigned to with its Hilbert space. Then you let those bits interact. And for a classical computer, that should be fairly independent. If I store a value of 0 here, this is independent if next to it is a 0 or a 1. For DNA, the value, the quantum information of a single site strongly depends on the quantum information on the next site. And for full quantum computing, entanglement seems to be the crucial quantity. So then you fully exploit the non-local correlations. With classical computation, you do gates like and, or, not, and many more. In quantum computing, you have a universal set of unitary gates. And I have to say that in DNA, I have no idea what sort of gates are possible. So it would be also interesting if to work together with someone in biology to actually classify the allowed operations on DNA. When it comes to cutting DNA and reassembling, that's getting pretty wild. And also, non-equilibrium will be the most interesting case. What I said about shaking one end and seeing how the correlation spread along the chain, that's non-equilibrium. The gate time will be short for classical and quantum computing. In classical computing, the shorter the gate time, the faster your processor works. For quantum computing, you always need to find the decoherence, so that's why you have to make it short. DNA will be a little bit different because you have this sort of continuous interaction. And then finally, the readout for a classical computer that's fully deterministic and you make only few errors. With quantum computing, that's actually statistical. You need to repeat the same experiment many times before you can actually have a readout. And DNA is, again, somewhere in the middle. So if you want to read out DNA information, you have a single shot. You have one try to do it. So given all the quantum that's involved there, I would actually like to know the accuracy of that readout. So there are many, many more things you could compare. But what you see already is that DNA seems to sit somewhere in the middle between classical and quantum computing. So the conclusions of this talk is that both classical and quantum information should be considered for full understanding of DNA, even if DNA is probably not a full quantum computer. And there are many people I would like to thank for helpful discussions for preparing this talk. So Mila Gu, Oscar Dalston, Kavan Modi, Giovanni Vacanti, Janet Anders, my supervisor, Vlatko Vetral, and there are probably many more. And yeah, thank you for your attention.
The first is, I would like to solve your paradox. Actually, there's no information gained. Yeah. Because you have to look at the entire environment, which includes water. And this is a typical problem in protein folding. When you include water, it's an entropic force. So you calculate all the energy, and you minimize free energy of the entire system, which is DNA with water or protein or peptides with water. And you end up with the equilibrium conformation, which is the folded state. And that corresponds to the minimum energy or maximum entropy. So I mean, of course, we have to do detailed calculations, but I maintain that when you do that, you will end up with possibly loss of information, not gain of information.
Of course, when you consider the whole box, then of course you cannot gain information. But still, I would like to understand how this information from the water molecules actually...
No, it's very simple, because water molecules attach to peptides or amino acids. And by doing so, they lose information about their conformational freedom of movement. So free water molecule has rotational degrees and translational. When it's bound, it loses this freedom. And that can be quickly calculated into entropy gain or information loss. The idea of the superposition of quantum and classical wave function for the DNA, I think it's a great one. And I think you call this shadow information or something. But actually, it can be made very concrete. We think about DNA as the sequence of nucleotides, the code. But actually, it's not letters. These are the molecules that they're vibrating. So the vibrations around the bonds to me are like wave functions. So this is really quantum mechanical oscillations, and that can be your shadow, if you will. Because if you think about just a sequence, yes, and these vibrations actually reflect the neighborhood. So what you talked about, the importance of the neighborhood, it's preserved by the nature of the vibrations. And finally, very quick, the gates and the readout. I think there's a whole slew of proteins like transcription factors and DNA polymerases, enzymes that participate in this, and these are the readers and the writers and the gates and all this. Anyway, that's all I have to say.
speak loud? Yeah. Okay.
So, did you mention at all why the quantum coherence could survive at room temperature for these DNA molecules?
Yes, so one part of the quantum information is encoded in this electronic degree of freedom, and it just happens that you have optical frequencies for this system, and your room temperature cannot excite those optical frequencies. So that's why you are basically in the ground state. Another thing is that you have this double helix, and that actually shields very well any noise from it.
So you are saying this has nothing to do with non-equilibrium state of this DNA vibrational mode. So you are saying even in a static form.
Yes. So far my calculations are only static, and in the next step I will look at non-equilibrium. But even in static, you have a massively stable system. Massively stable in the sense of maintaining quantum information because of those optical frequencies.
Yeah.
Well, with the proton tunneling, it's more delicate because you can only measure on one basis, and then it's actually undistinguishable whether you have a coherent superposition or a mixture. But for tunneling to take place at some point, it needed to be a superposition. That's why I denoted here. I'm unfortunately a physicist. I hope that some biologists in the audience can answer.
Yeah, because this goes to the comparison at the end that you did, the table that you compare, classical computing DNA and quantum computing. I mean, I don't see any discussions of the coherence time scales here. So I think
we can add it in the next coffee break.
Okay.
Let's take one more question, then we'll have a quick break.
I think there are lots of experiments we know about that we show entanglement over space. And I wonder if you're suggesting that there's entanglement over time with DNA sort of being the string between.
That's a slight misunderstanding from the notation I used. You can have entanglement over time. Here I do not propose it. It's that you have the classical information in the past, which you want to send into the future, and each step might use quantum mechanics, but that's a different problem.
Okay, I'd like to thank our speaker one more time. Thank you.